There's a number that oncologists do not lead with when they explain colorectal cancer to a newly diagnosed patient. The number is 89, as in 89% of colorectal tumors examined in a landmark study published in the journal Science contain high concentrations of a specific bacterium. A bacterium that, in healthy individuals, exists in the mouth and in extremely small quantities in the gut, causing no obvious harm and attracting no attention. A bacterium so unremarkable in its normal residence that most people reading this have never heard its name. A bacterium that, when it migrates, when it leaves the mouth, survives the acid of the stomach, colonizes the colon, and begins embedding itself into the tissue of the gut wall, appears to do something that no bacterial behavior had previously been observed to do at this scale in human cancer biology. It feeds the tumor. Not metaphorically, mechanistically. It provides the tumor with the precise molecular signals needed to evade the immune system, to resist chemotherapy, to recruit blood vessels that deliver it nutrients, and to suppress the very cellular programs that exist specifically to force cancer cells into self-destruction. And 89% of colorectal tumors have it. The bacterium is called Fusobacterium nucleatum. And the story of how it was discovered inside tumors, what it does once it gets there, and critically, what you can do about it is not a story most people with a gut have ever been told. But they should have been, because Fusobacterium nucleatum does not appear out of nowhere. It does not infect people randomly the way a cold virus does. It takes up residence in a specific gut environment. It thrives under specific conditions. It proliferates when certain things are present in the diet and the microbiome, and it is suppressed when others are. It is, in other words, not purely a matter of bad luck. I want to start somewhere unusual. Not with the bacterium. With the moment a researcher at Harvard looked at a genetic map of a colon tumor and found something that had absolutely no business being there. The year was 2011. Matthew Meyerson and his team at the Broad Institute were doing what genomic cancer researchers do. They were sequencing tumors, mapping the genetic landscape of colorectal cancer cells, looking for the mutations and alterations that distinguish cancer from normal tissue. Routine work by the standards of modern oncology. Important, precise, technically demanding, but conceptually familiar. What they found was not familiar. Buried in the sequencing data, alongside the expected cancer-associated mutations, was an enormous quantity of bacterial RNA. Not human. Not cancer. Bacterial. And not just any bacterial RNA, RNA from Fusobacterium nucleatum, an anaerobic bacterium that lives predominantly in the human oral cavity, found in the gums, in periodontal pockets, in the spaces between teeth, a mouth bacterium, inside a colon tumor. Meyerson described his initial reaction in a subsequent interview as confusion bordering on disbelief. The team ran the analysis again. The contamination possibility was exhaustively ruled out. The bacterium was not a contaminant. It was genuinely there, in the tumor, present in quantities far exceeding anything that could be explained by incidental transit through the digestive system. Within 3 years, more than 200 studies had examined the relationship between Fusobacterium nucleatum and colorectal cancer. The 89% figure came from a meta-analysis of these studies. And what emerged from this body of work was not a simple association, it was a mechanism. A detailed, multi-pathway, molecularly specific mechanism by which this bacterium actively promotes cancer growth. When Fusobacterium nucleatum colonizes the colon and encounters precancerous or cancerous cells, it does not simply sit alongside them passively. It has a surface protein called FADA, a specific adhesin that binds to E-cadherin, a protein on the surface of colorectal cells. E-cadherin is, under normal circumstances, one of the primary regulators of cell growth and cell-to-cell adhesion. It keeps cells behaving as part of an organized tissue rather than as individual rogue entities. When FADA binds to E-cadherin, it triggers a signaling cascade inside the cell. Specifically, it activates beta-catenin, the same downstream effector of the WNT pathway we have discussed in the context of stem cell biology. In a cancer cell, activated beta-catenin is not doing what it does in a healthy stem cell. It is activating a completely different suite of genes. Genes that drive cell proliferation, genes that suppress apoptosis, which is the programmed cell death that is the body's primary defense against cancer, and genes that increase the cell's invasive capacity. In plain language, Fusobacterium nucleatum grabs the surface of a cancer cell, pulls a molecular lever, and tells the cell to grow faster, stop dying, and spread. But fata and beta-catenin are only the beginning. Fusobacterium nucleatum produces a toxin, a protein called FAMA, that directly suppresses natural killer cells and cytotoxic T cells, the immune cells specifically tasked with identifying and destroying tumor cells. The immune system, without this suppression, would recognize most pre-malignant cells and eliminate them before they became tumors. It does this successfully thousands of times per day in a healthy individual. The reason some cells escape this surveillance and become tumors is not that the immune system is completely blind. It is that the cancer cells and their microbial accomplices are actively suppressing the surveillance system. Fusobacterium nucleatum recruits myeloid-derived suppressor cells, MDSCs, into the tumor microenvironment. MDSCs are immune cells that, in their normal context, suppress immune responses to prevent excessive inflammation. Inside a tumor, they suppress immune responses to prevent the killing of cancer cells. Fusobacterium nucleatum specifically signals for MDSC recruitment. It builds the immunosuppressive environment around itself and the tumor as an act of self-preservation. And then there is the chemotherapy resistance finding, which I want to dwell on because it has direct clinical implications that are still not widely communicated to patients receiving treatment. In 2017, a study published in Cell Host and Microbe, conducted by Wendy Garrett's laboratory at Harvard, demonstrated that Fusobacterium nucleatum present in colorectal tumors confers resistance to two of the most commonly used chemotherapy regimens for colorectal cancer, oxaliplatin and 5-fluorouracil. The mechanism was through activation of autophagy, the cellular recycling program, in tumor cells. Normally, autophagy in cancer cells is a double-edged sword, sometimes promoting and sometimes suppressing tumor growth. What the Garrett laboratory found was that Fusobacterium nucleatum activates a specific selective autophagy pathway in tumor cells that degrades the chemotherapy drug before it can act. The cancer cells are literally eating the medicine, and Fusobacterium nucleatum is teaching them how. Patients with high Fusobacterium nucleatum burden in their tumors showed significantly worse outcomes on standard chemotherapy compared to patients with low burden. The bacterium was not just feeding the tumor during its growth phase, it was protecting it during its treatment phase. Read that again. A bacterium protecting a tumor from chemotherapy in 89% of colorectal tumors examined. High Fusobacterium nucleatum colonization of the colon is associated with poor oral hygiene, specifically periodontal disease, which provides a reservoir of Fusobacterium nucleatum in the gums that can be swallowed continuously. It is associated with high intake of processed meat and red meat, which in the colon produces heme-iron in a form that promotes an anaerobic, low-oxygen environment that Fusobacterium nucleatum thrives in. It is associated with low dietary fiber intake, which reduces the butyrate-producing bacteria that acidify the colon environment and suppress Fusobacterium nucleatum's growth. And it is associated with alcohol consumption, which increases gut permeability and disrupts the mucus layer that normally acts as a barrier preventing bacterial access to the colonic epithelium. I want to pause here and say something directly. This is not a claim that diet prevents cancer. The relationship between the gut microbiome, Fusobacterium nucleatum, and colorectal cancer involves genetic factors, immune factors, environmental exposures, and elements of biology that remain incompletely understood. What I am describing is a modifiable aspect of a complex picture, a significant one, backed by substantial evidence, but one component among several. What I am saying is that Fusobacterium nucleatum is a real, mechanistically understood contributor to colorectal cancer initiation and progression. That its colonization of the gut is significantly influenced by dietary and oral hygiene factors that are within your control. That the evidence for specific interventions, food-based and behavioral, is specific enough and strong enough to be worth communicating. And that the failure to communicate it broadly is a gap in public health that leaves people without information they deserve to have. With that established, let me tell you what the research actually supports. There is a person I want to introduce you to here. Not by name, I will call him Marcus. He came to me not as a patient in the clinical sense, but as someone who had received a diagnosis and was trying to understand, with the same systematic intelligence he had applied to a long career as an engineer, exactly what was happening in his body and what he could actually do about it. Marcus was 61. He had been diagnosed with stage 2 colorectal cancer 8 months before we spoke. He had completed surgery and was partway through a chemotherapy regimen. What he wanted to know, and what he was not getting from the clinical appointments that were necessarily focused on treatment protocols and monitoring, was what the tumor biology actually looked like. What was driving it? Whether anything he was doing was helping or hindering the treatment. He had read the Fusobacterium nucleatum literature, not the popular press coverage, the original papers. He had, in the methodical way of someone accustomed to reading technical specifications, gone through the cell host and microbe study, the science papers, the subsequent mechanistic work, and he arrived with a very precise set of questions. He wanted to know whether his tumor had been tested for Fusobacterium nucleatum presence. It had not. This is not routine clinical practice, and in most oncology centers worldwide, it remains a research finding rather than a clinical test. He wanted to know whether his diet was creating a gut environment that could be supporting residual bacterial colonization. This question was more answerable. And he wanted to know whether specific interventions could measurably shift the microbial balance in his colon during treatment. The answer to the last question is yes, and the evidence is specific enough to describe precisely. The first intervention that the research supports most strongly is the modulation of gut pH through dietary fiber fermentation. Fusobacterium nucleatum is an anaerobic organism that tolerates acid but does not thrive in it. The short-chain fatty acids produced by fermentation of dietary fiber, butyrate, propionate, acetate, acidify the colonic environment and create conditions that suppress Fusobacterium nucleatum growth. Butyrate specifically does something additional that makes it doubly important in this context. It is the primary fuel source for colonocytes, the cells lining the colon. Well-fueled colonocytes maintain the tight junctions and the mucus layer that prevent bacterial translocation from the gut lumen into the colonic wall. When butyrate is adequate, the mucus barrier is thick and intact. When butyrate is low, as it is in people eating low-fiber Western diets, the mucus barrier thins and bacteria like Fusobacterium nucleatum gain access to the epithelium. The fatty adhesin can only do its damage when the bacterium reaches the colonic wall. A robust mucus layer is the first physical barrier preventing that access. Butyrate maintains that barrier. The foods that generate the highest butyrate production in the human colon are not complicated or exotic. They are the foods that have been the foundation of traditional diets across every culture that has maintained lower colorectal cancer rates than the modern Western world. Cooked and cooled legumes, which develop resistant starch on cooling, raw oats, cooked and cooled potatoes, leeks, onions, garlic, chicory root, and Jerusalem artichokes. These foods provide inulin, pectin, arabinoxalan, and resistant starch, the fermentable substrates that butyrate-producing bacteria, particularly Faecalibacterium prausnitzi and Roseburia intestinalis, convert into butyrate. The second intervention is one that surprises almost everyone when I describe it, and yet the evidence for it is among the most specific in the entire Fusobacterium nucleatum literature: garlic. Specifically, a sulfur compound in garlic called allicin and its derivative compound ajoine, which forms when raw garlic is crushed or chopped and allowed to sit for 10 to 15 minutes before cooking or consumption. Allicin and ajoine have been demonstrated in direct antimicrobial testing to inhibit Fusobacterium nucleatum adhesion to colonic epithelial cells with remarkable potency. The mechanism is specific. Allicin binds to and inactivates the fatty adhesin directly. The grappling hook that Fusobacterium nucleatum uses to attach to E-cadherin on colonic cells is disarmed by this sulfur compound. A 2019 study in Frontiers in Microbiology tested multiple dietary compounds against Fusobacterium nucleatum in conditions designed to mimic the colonic environment. Allicin was among the most potent inhibitors identified. The effective concentration, the amount needed to meaningfully inhibit adhesion, was achievable through dietary consumption of 2 to 3 cloves of raw or lightly cooked garlic per day. Marcus began eating raw garlic. Initially with significant reluctance, because the social implications of raw garlic consumption in professional settings were something he had opinions about. We found a workable middle ground. Crushed garlic rested for 12 minutes, then added to olive oil and poured over a salad or vegetables at room temperature. Adequate allicin delivery, considerably more socially manageable. The third intervention connects directly to the oral hygiene finding, the one that is perhaps the most uncomfortable because it implicates a part of the body that most people do not connect to gut cancer at all. Fusobacterium nucleatum is an oral bacterium. It lives in the gingival crevice, the space between the gum and the tooth, and in periodontal pockets, the deeper spaces that form when gum disease causes the gum to pull away from the tooth surface. In people with periodontal disease, Fusobacterium nucleatum populations in the mouth can be orders of magnitude higher than in people with healthy gums. The route from mouth to colon is simple: swallowing. Every person swallows approximately 1 to 1.5 liters of saliva per day. In a person with periodontal disease and high oral Fusobacterium nucleatum burden, that swallowed saliva contains a continuous stream of bacteria making the journey downward. Most are destroyed by stomach acid, but Fusobacterium nucleatum has demonstrated acid tolerance. It survives gastric transit at rates sufficient to establish colonic colonization, particularly in people whose gastric acid production has declined with age. After age 60, gastric acid production falls in a significant proportion of people. The selective barrier that stomach acid provides against oral bacteria reaching the colon weakens. And in people on proton pump inhibitors, medications that further suppress gastric acid, taken by hundreds of millions of people worldwide for reflux, that barrier is functionally very limited. The connection between periodontal disease and colorectal cancer has been studied epidemiologically, and the findings are sobering. A 2018 meta-analysis in the European Journal of Cancer found a significantly elevated colorectal cancer risk in individuals with documented periodontal disease, consistent across multiple independent cohort studies spanning different countries and populations. This does not mean gum disease causes colon cancer. The relationship is multifactorial, but Fusobacterium nucleatum connects the two in a mechanistically coherent way. Periodontal disease is the reservoir, swallowing is the transport mechanism, gastric acid insufficiency is the vulnerability, and colonic colonization is the outcome. The implications for daily practice are straightforward. Thorough toothbrushing twice daily, including brushing the gum line rather than just the tooth surface. Daily flossing, specifically to disrupt the bacterial biofilm in the gingival crevice where Fusobacterium nucleatum concentrates. Regular professional cleaning, which removes the calcified plaque, calculus, that harbors Fusobacterium nucleatum in protected pockets that brushing cannot reach. And for people on long-term proton pump inhibitors, a conversation with their physician about whether the lowest effective dose is being used and whether alternatives exist. The fourth area of intervention involves something that is perhaps counterintuitive, not what to add, but what to remove. And the removal is of a specific dietary pattern rather than a specific food. Processed red meat, bacon, sausages, ham, hot dogs, deli meats, creates a specific colonic chemistry when digested. The heme ion in red meat, when processed in the colon, produces reactive oxygen species and promotes the formation of N-nitroso compounds, which are directly genotoxic, meaning they damage DNA in colonic cells. But the impact on Fusobacterium nucleatum specifically comes through a different pathway. The fermentation of protein by colonic bacteria, specifically the protein that is not absorbed in the small intestine and reaches the colon intact, which is higher with processed meat consumption, produces putrefactive metabolites including hydrogen sulfide, indoles, and phenols. These compounds shift the colonic environment toward alkalinity and toward anaerobic conditions that specifically favor Fusobacterium nucleatum over the beneficial acid-producing bacteria. High processed meat intake is essentially creating the environmental conditions that roll out the welcome mat for exactly the organism you do not want colonizing your colon. The intervention here is not eliminating all meat. The evidence specifically implicates processed red meat, not unprocessed red meat consumed in modest quantities, and not white meat or fish. The practical change is removing the daily processed meat habit, the bacon at breakfast, the deli ham in the sandwich, the sausage several times a week, and replacing those protein sources with fish, legumes, eggs, or unprocessed poultry. The fifth intervention brings us to something that has appeared repeatedly across the research into gut health and cancer biology, and that keeps showing up for good mechanistic reasons. fermented foods, and the specific bacterial competitors of Fusobacterium nucleatum they introduce. Lactobacillus strains, the dominant bacteria in traditionally fermented foods like plain kefir, raw sauerkraut, kimchi, and live culture yogurt, are direct competitors of Fusobacterium nucleatum in the colonic environment. They produce lactic acid and hydrogen peroxide as metabolic byproducts, both of which are inhibitory to Fusobacterium nucleatum growth. They compete for adhesion sites on the colonic epithelium, physically displacing Fusobacterium nucleatum from the colonization niches it would otherwise occupy. And the lactic acid they produce contributes to the colonic acidification that, alongside butyrate from fiber fermentation, creates an environment that selectively disadvantages anaerobes like Fusobacterium nucleatum. A 2020 study published in Gut Microbes demonstrated that Lactobacillus acidophilus and Lactobacillus rhamnosus, both present in high-quality kefir and fermented dairy products, significantly reduced Fusobacterium nucleatum adhesion to colonic epithelial cells in vitro and reduced Fusobacterium nucleatum-driven activation of beta-catenin signaling, the exact pathway through which the bacterium promotes cancer cell proliferation. 1 cup of plain, full-fat kefir daily. 2 tablespoons of raw, unpasteurized sauerkraut at 1 meal, both together if possible. The bacterial diversity these two foods deliver works synergistically. Kefir predominantly colonizing the small intestine, sauerkraut organisms more active in the large intestine, together providing competitive coverage across the full length of the gut. There is a 6th area that the research has begun to illuminate in the last 4 years, and I want to describe it because it connects everything else in a way that the individual interventions do not fully convey on their own. The relationship between Fusobacterium nucleatum and the rest of the gut microbiome is not simply competitive. It is ecological. Fusobacterium nucleatum does not arrive in a vacuum and do damage in isolation. It arrives in a gut environment that is either welcoming or hostile, and that environment is a product of the entire community of bacteria present, their diversity, their metabolic output, their competition for resources and adhesion sites. Akkermansia muciniphila is the most discussed of these. It is a bacterium that lives in and maintains the gut mucus layer, producing enzymes that recycle mucus components and stimulating goblet cells to produce more mucin. A thick, intact mucus layer is, as we established, the first physical barrier preventing Fusobacterium nucleatum from reaching the colonic epithelium. Akkermansia keeps that barrier robust. A gut with abundant Akkermansia is a gut with a better physical defense against the bacterial adhesion that initiates the cancer-promoting cascade. Akkermansia muciniphila is fed by a specific prebiotic substrate, polyphenols. Particularly the polyphenols in pomegranate, elagitannins, dark berries, and green tea catechins. These compounds are not directly absorbed in the small intestine in most people. They pass through to the colon where they selectively feed Akkermansia and other beneficial mucosal bacteria while being inhibitory to pathogenic organisms, including Fusobacterium nucleatum. Green tea deserves specific mention here because its catechin profile, particularly epigallocatechin gallati, EGCG, has been studied directly against Fusobacterium nucleatum, with results that are difficult to overstate. A 2018 study found that EGCG inhibited Fusobacterium nucleatum biofilm formation, the structured community that makes the bacterium more resistant to elimination, at concentrations achievable through regular green tea consumption. 3 to 4 cups of unsweetened green tea per day, brewed for 3 to 5 minutes at below boiling temperature to preserve catechin content, provides the relevant EGCG dose. The green tea also feeds Akkermansia. The Akkermansia maintains the mucus barrier. The mucus barrier prevents Fusobacterium nucleatum adhesion. The allicin from garlic disarms the fada adhesin directly. The butyrate from fiber acidifies the colonic environment. The lactobacillus from fermented foods displaces and directly inhibits the bacterium. The reduction of processed meat removes the environmental conditions that favor its proliferation. The oral hygiene reduces the source reservoir. These interventions are not a random collection of healthy behaviors dressed up in mechanistic language. They are a specific, layered response to a specific, mechanistically understood threat, each acting at a different point in the pathway by which Fusobacterium nucleatum colonizes the colon and promotes cancer growth. Marcus asked me toward the end of our conversations whether there was any clinical measure that could tell him whether all of this was actually working. Whether, beneath the improvement in how he felt and the normalization of his bowel function, the microbial landscape of his colon was actually changing in the way the research predicted. The answer is yes, with caveats. Fecal microbiome testing, available through several commercial laboratories as well as through research programs, can measure the relative abundance of Fusobacterium nucleatum DNA in a stool sample, expressed as a proportion of the total bacterial DNA present. It is not a perfect test, and the interpretation requires some caution because day-to-day variation is real and single samples are imperfect snapshots. But across 3 to 6 months of consistent dietary change, the directional shift is detectable. Marcus tested at baseline and again at 5 months. His Fusobacterium nucleatum relative abundance had fallen from the 94th percentile compared to reference populations to the 31st percentile. Faecalibacterium prausnitzi, the butyrate-producing bacterium that had been nearly absent from his initial sample, had risen to the 73rd percentile. These are not published clinical trial results from a controlled study. They are one person's data from a home microbiome test, and I present them as illustration rather than evidence. But they are consistent with what the mechanistic research predicts would happen given the interventions he made. And they gave Marcus something that the tumor board appointments and the chemotherapy monitoring visits could not give him: a sense of agency over a part of his biology that had, up until that point, felt entirely beyond his influence. I want to close with something that goes beyond the specific bacterium and the specific interventions because I think the broader point is worth making explicitly. The discovery of Fusobacterium nucleatum inside colorectal tumors and the subsequent decade of research uncovering what it does and how it gets there represents something genuinely new in our understanding of cancer. Cancer has been framed for most of its modern clinical history as a disease of genetics, of mutations, of DNA errors, of cellular programs gone wrong from within. The microbiome research is revealing something more complex and, in an important sense, more hopeful, that the environment surrounding cancer cells, including the microbial environment of the gut, is not a passive bystander. It is an active participant. It can be tilted toward tumor promotion or toward tumor suppression. And the levers that tilt it are, to a meaningful degree, within the daily choices of the person who carries it. This is not an argument against oncology as it exists. Surgery, chemotherapy, immunotherapy, targeted therapy, these are not rendered obsolete by a conversation about garlic and kefir. They remain the primary interventions for treating established cancer. What the Fusobacterium nucleatum research adds is not a replacement for those interventions, but an additional layer of biological understanding that has direct implications for prevention, for adjunct support during treatment, and for the reduction of recurrence risk after treatment. Your gut microbiome is not fixed. It responds relatively quickly, within weeks for the most responsive changes, within months for deeper shifts in colonization, to changes in what you eat, how you maintain your oral health, and which specific bacteria you introduce through fermented foods. The balance between the bacteria that feed tumors and the bacteria that suppress them is not determined solely by genetics or by chance. It is, in significant part, determined by the environment you create in your colon every single day. High-fiber food, 35 grams per day minimum, legumes, cooked and cooled resistant starch, leeks, onions, garlic, raw oats. 2 to 3 cloves of garlic daily, crushed and rested for 10 minutes. 1 cup of plain kefir in the morning. 2 tablespoons of raw sauerkraut at lunch or dinner. 3 to 4 cups of unsweetened green tea. Pomegranate seeds or concentrate or mixed dark berries daily. Fatty fish 3 times per week. The elimination of daily processed and preserved meat. Thorough oral hygiene, twice daily with flossing and professional cleaning on schedule. The 12-hour overnight fasting window that allows the colonic microbiome to complete its repair cycle overnight without competing with incoming food. None of these are dramatic. None of them are expensive. Several of them are foods that traditional populations with low colorectal cancer rates have eaten as dietary staples for centuries without knowing the molecular reason, but observing across generations that something about how they ate kept something at bay. We know the molecular reason now. The bacterium is real. What it does is real. The conditions that invite it are real. And the conditions that suppress it are real. You have the information now. Use it.